Welcome to the Deep Dive, the place where we take the densest research, the latest breakthroughs, and the most complex data and really boil it all down. We aim to give you the most surprising and essential knowledge you need without drowning you in jargon. Today, we are peering through the microscope, really focusing in on one of the most common, yet, let's face it, frustrating staples of pharmacy R&D, the simple eye drop. But we're not talking about your basic, over-the-counter saline solution, not at all. We are doing a highly technical deep dive into the next generation of ophthalmic formulations. This is the cutting edge of drug delivery, designed for maximum patient comfort. Our sources show a fundamental shift happening in pharmaceutical R&D. They're moving quite aggressively, actually, away from those irritating synthetic chemicals. They're
Yeah.
moving toward solutions that are, well, essentially biomimetic. They're designed to flawlessly mimic your body's own chemistry. The idea is correcting problems without causing new ones.
That's exactly it. That's the crux of the challenge right now, and it's enormous for anyone in formulation science. The industry is fundamentally shifting away from those, let's call them traditional emulsions, which often rely, pretty heavily on strong synthetic surfactants just for basic stability. You can still see these sort of legacy formulation approaches in some current commercial products, like certain emulsified sprays from companies such as Boy. They work, they're functional, but they still incorporate components that, well, they compromise comfort. The new focus, the non-negotiable standard for the future, is developing physiologically compatible nanotechnological solutions. These are built from the ground up. The whole idea is to bypass the chronic toxicity issues that have really plagued eye care for decades, especially for long-term treatments.
Okay, so if comfort and safety are the big goals, what's the central mission here? What are they actually trying to achieve beyond just, saying, hey, these drops feel nicer?
Right, it's more than just nicer. The core mission is multifaceted, but it really boils down to achieving two specific things at the same time. First, and this is crucial, the development of totally preservative-free formulations. This isn't a minor detail, it's basically non-negotiable now. The goal is to eliminate all chemically induced toxicity that comes from those antimicrobial agents that used to use for shelf stability. Second, maximizing patient compliance by tailoring the delivery method. So these next generation treatments need to work either as low viscosity sprays, which are comfortable for easy sort of intermittent daytime use, or as higher viscosity gels and ointments, which are optimized for, say, overnight retention.
And there's a really critical physical constraint too, isn't there? Regardless of whether it's a spray or a gel, the actual particles inside have to be incredibly tiny.
Absolutely. That's the initial physical hurdle that really dictates engineering success. The particles within that liquid matrix, they must remain precisely within the nanoscale. We're talking about particles that range, ideally, somewhere between 20 and 200 nanometers.
20 to 200 nanometers, that's unbelievably small.
It is, and if those particles start to aggregate or, coalesce and grow significantly larger than 200 nanometers, they immediately cause optical scatter. That leads directly to blurry vision. Plus, they create a kind of grittiness or discomfort on the ocular surface, and that just defeats the entire purpose of a comfort-focused, high-tech formulation, right?
Right, so the chemistry could be perfect, but if the particle size is off, the whole product fails.
Exactly. The physics has to be right, too.
And the stakes here are incredibly high. We're not just talking about minor eye irritation, are we? We're talking about chronic debilitating conditions.
Precisely. This shift in chemical R&D, it's critical for managing chronic severe issues. We're discussing conditions like dry eye disease, DED, and meibomian gland dysfunction, MGD. These affect millions of people globally. Current traditional solutions for these conditions, they often force patients into a really difficult trade-off. They have to compromise on comfort to get the necessary stability or the drug efficacy they need. The new chemical framework demands that we provide maximum therapeutic efficacy without that chemical compromise. And that means solving the problem of irritation first. It has to be the priority.
Okay, let's unpack this then. Before we can build this perfect, comfortable eye drop of the future, we really need to understand the core failure of the past. Why exactly did those old formulations cause pain? You mentioned it's not just a physical discomfort thing, it's a direct molecular chemical event, almost like a cellular attack.
That's probably the most accurate way to put it, yeah. To innovate successfully, we first have to define the molecular mechanism of irritation. That way, we know precisely which chemistries to exclude and, just as importantly, which ones to replace them with. The fundamental problem lies with the detergent properties that are inherent in many ingredients traditionally used for stability and preservation.
Detergent properties, like soap.
Essentially, yes. Think about the common ophthalmic preservatives compounds like benzalkonium chloride, BAK, cetramide, or acetylpyridinium chloride. Why are they so effective at killing microbes and preventing contamination in the bottle? Because they are strong cationic surfactants. They're basically detergents. Their function is to disrupt and compromise the integrity of cell membranes. Microbial cell membranes, yes, that's the goal. But they do the exact same thing to the very delicate epithelial cells on your ocular surface.
Ah, I see.
They work by literally pulling the lipids out of the cell membrane, creating holes, and eventually causing the cell to break apart or lyse. So that mechanism, the very thing that keeps the product stable in the bottle, is inherently cytotoxic to the eye itself.
Wow. So the preservative is literally dissolving the outer layer of the eye cells, causing damage, and that's what we feel as chemical irritation and inflammation.
Exactly. And the clinical world has definitely recognized this toxicity. It's not subtle. This is why there's such a strong, ongoing clinical shift toward preservative-free eye drops, and specifically toward drops that incorporate lipids. The purpose of adding lipids is kind of twofold. First, they provide a protective, replenishing layer to the tear film, which is often compromised in dry eye. And second, using lipids helps minimize or even eliminate the need for those traditional synthetic surfactants and those harsh detergent preservatives.
So even if they tried to hide the preservative, say, inside an oil droplet.
Right, even if a formulator tries to encapsulate a preservative like BA within an oil phase to reduce direct exposure,
Yeah.
that compromise is still considered unacceptable in this high-comfort, preservative-free future we're aiming for. It's just not good enough anymore.
Okay, let's drill down into that specific pain trigger. You mentioned this isn't just some vague burning sensation. There's a specific receptor reacting to the chemical structure of these detergents. What is that receptor, and how does it translate a chemical presence into an actual pain signal?
This is really where the chemistry informs the comfort directly. The molecular mechanism of irritation centers on a specific nociceptor that's a pain receptor called TRPV1. TRPV1 stands for transient receptor potential phenyloid 1. And these receptors are densely packed on the sensory nerve endings right on the surface of your eye.
TRPV1.
Yeah, TRPV1 is fascinating because it's highly sensitive to various irritants, things like noxious heat, low pH, and importantly for us, chemical irritants. When an irritant chemical, in this case, a detergent surfactant, binds to that receptor, it acts like a molecular key. It changes the shape, the conformation of the channel protein and physically opens up this ion channel. For the organic chemists listening, it's critical to understand this is a direct, dose-dependent molecular interaction. It causes an immediate signaling cascade.
Can you give some examples? What common chemicals that we might recognize actually trigger this TRPV1 pain response in eye drops?
Sure. We look at widely used anionic detergents like sodium lauryl sulfate, SLS. It's famous for creating foam in shampoos and body washes, right?
Right, SLS, I know that one.
Yeah, and also agents like cocamide monoethanolamide, or CMEA. Both of these are well-known activators of the TRPV1 receptor, and it happens in a dose-dependent manner. So the higher the concentration of the surfactant, the more these channels open up, and the more intense the sensation of pain and irritation becomes.
So the burning feeling we might get from some older drops is literally like putting microscopic soap onto our corneal nerve endings, setting off a very specific pain alarm. And what happens inside the cell once that TRPV1 channel is forced open by these surfactants?
Okay, so when the channel opens, it allows a massive, really rapid influx of calcium ions, that's Nathan Kip 2 plus other, from the fluid outside the cell into the cell's cytoplasm. This abrupt flood of Matham key 2+ is the fundamental chemical signal that's directly linked to transmitting the pain sensation back to the brain. And beyond just the immediate pain, this calcium overload can trigger secondary inflammatory responses and cellular stress. If the exposure is chronic, like using drops every day, this can lead to long-term epithelial damage.
That really underlines the need to go completely preservative-free. It sounds absolutely critical. You mentioned a crucial contrast in the sources about cell viability. Could you walk us through that specific example? It really highlights the difference between detergent-based and truly biocompatible formulations.
Yes, the contrast is incredibly stark, and it provides the solid scientific justification for this whole new R&D direction. When researchers took an otherwise promising liposomal formulation, a good base structure, and they incorporated a strong detergent preservative like BAK, the result in cell culture studies was devastating. It led to the complete and irreversible loss of epithelial cell viability.
Complete loss. The cells just died.
The cells died. The detergent structure was simply too potent, too disruptive. Now, conversely, they took a formulation that was similarly structured based on phosphatidylcholine liposomes, but was rigorously preservative-free. That formulation allowed for immediate cellular recovery after exposure and maintained long-term cell health. So the presence or absence of just that single detergent molecule, even potentially in trace amounts, defines whether the epithelial cell lives and recovers or whether the formulation is inherently toxic. This single finding is really the foundation of the chemical exclusion strategy we're seeing now. Exclude the toxins entirely.
Okay, so building on that, besides just excluding the toxic chemicals, the sources also emphasize that we have to stick to strict physicochemical boundaries. We need to mimic natural tears almost perfectly. Getting the physical chemistry of the liquid environment exactly right seems just as important for comfort. Let's tackle these constraints one by one.
Absolutely. If the physical environment is wrong, even the most perfect nanochemistry is going to fail on the comfort front. We have to achieve maximum biocompatibility by closely matching the natural tear film. The first constraint is pH. The optimum pH for maximum comfort, the point that minimizes chemical irritation, is approximately 7.4.
That's 7.4, okay.
That precisely matches the normal, healthy tear film pH. Now, the eye can actually tolerate a pretty wide range of pH, maybe from about 3.0 up to 8.6 without permanent damage. But the comfortable zone where it feels good is much narrower. Generally, we're talking a range from about 6.0 to 7.4.
So below 6.0 starts to sting.
Exactly. If the formulation's pH drops too low, it can cause immediate stinging irritation. And that's partly by activating those pH-sensitive components of the same TRPV1 receptor we just discussed. So the buffering capacity of the formula is critical. It needs to maintain that narrow, comfortable pH window even when it mixes with tears on the eye surface.
Right, because if it's not buffered well, the simple act of putting the drop in could shift the pH enough to trigger that irritation.
Precisely. The product has to be resilient against those minor changes in tear film chemistry upon application.
Okay, next up is osmolality. This refers to the concentration of solutes in the liquid, right? And this seems key for treating dry eye disease specifically, because DED itself often involves the wrong osmolality in the patient's existing tears.
That's spot on. The ideal osmolality for comfort and cellular health is isotonically around 300 millimoles per kilogram, or mathrm osm. This matches the natural balance of water and salts inside and outside the cells. It keeps them happy. The problem with dry eye disease often is that due to excessive evaporation or maybe a lack of the aqueous component in the tears, the remaining tear film becomes significantly concentrated. This is a state we call hypertonicity. The osmolality goes well above that ideal $300 mathrm osm care.
And that's bad for the cells.
It's intrinsically damaging, yes. This hypertonic state causes the corneal epithelial cells to actually shrink as water leaves them, and it stresses them severely. So DED treatment isn't just about adding fluid or lubrication. It's about correcting the existing hyperosmolality. We do that by applying a solution that is isotonic, around 300, or maybe even slightly hypotonic, below 300. The goal is to draw the tear film back toward that comfortable $300 mathrm osm cal range, reducing the cellular stress caused by the disease itself.
So if a patient's tears measure, say, 380 nolars mathrm ospicar, the therapeutic drop is actively trying to dilute or balance that concentration back down towards 300. That makes sense. It's reducing the stress.
Precisely. We're aiming for a therapeutic correction of the ocular surface environment, not just temporary relief.
Okay, and finally, the classic challenge, viscosity. We want something that stays on the eye long enough to work, but we don't want it to feel like glue or smear our vision. How does formulation science tackle this inherent trade-off?
Ah, yes, viscosity. It's the classic optimization puzzle in ophthalmic delivery. You're right. High viscosity is great because it increases the retention time, how long the drop stays on the precorneal surface. And that dramatically improves drug bioavailability. More contact time, more absorption. But the moment viscosity gets too high, it leads to unacceptable discomfort, that smeary vision, sticky eyelids. Patients hate it.
So what's the trick?
The secret, which the sources really highlight, is moving away from simple, Newtonian fluids where viscosity is constant, towards fluids that exhibit pseudoplastic flow behavior. It's also known as shear thinning. This is really a material science breakthrough for eye drops.
Okay, explain pseudoplastic flow for us. What does shear thinning actually mean for the patient experience?
It means the fluid acts thin. It has a low viscosity when a shear stress is applied to it, like the force generated when you blink, or even the force from applying a spray. Under that stress, the polymers in the formulation temporarily align themselves, reducing friction, making it flow easily. But the moment the eye is at rest and the shear stress drops, like between blinks, the polymers re-entangle, they relax, and the fluid thickens up again.
Ah, clever. So it's thin when moving, thick when still.
Exactly. This maximizes retention when you need it while the eye is open and the drop is just sitting there. but it minimizes irritation during the blink cycle because it thins out. The sources cite a value like $2.45 channel acid mantle as a functional viscosity benchmark under shear. But the real key is the ability of the formulation to change its physical state in response to mechanical input. It's an engineered responsiveness that lets us have both comfort and retention, which was always the tricky part.
That pseudoplastic flow is a fantastic example of smart engineering solving a physical problem. Now, let's pivot back to the chemistry side. If detergents are the big problem, if we absolutely have to avoid anything that triggers that TRPV1 receptor, how do we keep water and oil together stably? Because the tear film itself is the sophisticated mix of water and lipids that needs to stay emulsified. The answer, you mentioned earlier, lies in a tiny self-assembling bubble.
The answer is liposomes. And yes, they are essentially the perfect chemical antidote to the detergent problem. They achieve stability and encapsulation using natural components, but crucially, without the inherent toxicity of those synthetic surfactants we need to avoid. To understand them, you have to think chemically. A liposome is basically a microscopic vesicle system. It consists of an aqueous core like a tiny droplet of water or maybe a drug solution surrounded by a precisely engineered phospholipid bilayer.
A bilayer, like a cell membrane.
Exactly like a cell membrane. And this structure isn't forced together with harsh chemicals. It forms spontaneously in water. It's driven by a powerful natural phenomenon called the hydrophobic effect.
Okay, walk us through the hydrophobic effect in this context. How does it make these bubbles form?
Right. So phospholipids are amphiphilic molecules. That means they have two distinct parts. They have hydrophilic heads, which are water-loving phosphate-based groups, and they have hydrophobic acyl chains, long fatty acid tails that are intensely water-hating. When you put these lipids into a large volume of water, the hydrophobic effect dictates that those water-hating tails must turn away from the water phase to minimize contact. It's thermodynamically unfavorable for them to touch water. The most stable way, the lowest energy way for them to do this is to self-assemble into a closed spherical structure. The tails they tuck safely away inside the bilayer, shielded from water, and the hydrophilic heads face outward, interacting favorably with the aqueous environment. This self-assembling nature creates natural stability and completely bypasses the need for synthetic emulsifiers or detergents. So the chemistry itself forces the structure into existence. And that structure isn't just a container, right? You said the phospholipids themselves are actually beneficial. They're not just passive emulsifiers. Absolutely crucial point. These phospholipids, or PLs, common ones are phosphatidylcholine, PC, and phosphatidylethanolamine, PE, are naturally occurring components. They form the critical lipid layer of a healthy tear film. This isn't just structural support they provide. In dry eye disease caused by meibomian gland dysfunction, or MGD, we know there are well-documented deficiencies in specific lipids in the tear film, particularly PE and another one called sphingomyelin, SM.
So the liposomes are like delivering replacement parts.
Exactly. By introducing liposomes made from these very same phospholipids, we are designing formulations that actively replenish the tear film's lipid layer. They provide these essential natural building blocks, fundamentally improving the quality and stability of the tear film itself, specifically addressing that problem of excessive evaporation in DED.
Okay, so they form naturally, they're beneficial, but how do we keep these individual liposome bubbles from crashing into each other and clumping up? Especially considering we need them to stay stable on a shelf for maybe a year or two. Aggregation sounds like a big problem.
It is. That goes back to colloidal stability, keeping the particles dispersed. And it hinges significantly on the electrical charge of the lipid head groups. We pay close attention to whether the hydrophilic head group is negatively charged, positively charged, or zwitterionic.
Euterionic.
Yeah, zwitterionic means it carries both positive and negative charges simultaneously, but overall it might be neutral or have a very slight charge, depending on the pH. Phosphatidylcholine, PC, is a classic example.
Yeah.
This charge, or lack thereof in a specific way, is essential because it provides electrostatic repulsion between adjacent particles. Imagine two tiny spheres, both carrying a net negative charge, or both being zwitterionic with similar charge distributions. They naturally push away from each other, like trying to push the same poles of two magnets together.
Okay, like charges repel.
Exactly. This repulsive force prevents them from aggregating or clumping together over time, which is absolutely essential for long-term shelf stability and ensuring the particle size remains consistently within that comfortable, non-blurry 2200 millimeter range we need.
Liposomes are one structure, but the sources also talk about phospholipid nanoemulsions, or PNEs. How are these different from liposomes, and why are they also considered a breakthrough?
Right, a PNE is a variation, but it still adheres to the core principle of using biomimetic, non-irritating components. So a liposome, remember, is an aqueous core surrounded by a lipid shell, a bilayer. A nanomulsion is typically different. It's a tiny droplet of oil dispersed within a continuous water phase, like salad dressing, but on a nanoscale.
Okay, oil and water.
Yes. Now, traditional nanomulsions must use synthetic surfactants to stabilize that oil and water mixture and keep the tiny oil droplets from merging. And those surfactants are often the very TRPV1 activating irritants we're desperately trying to avoid. PNE systems are different. They specifically use the biocompatible phospholipids, PCPE, etc., as the primary and often the only emulsifiers.
Ah, so they replace the synthetic surfactant with natural phospholipids.
Exactly. This eliminates the need for any synthetic, potentially pain-inducing agents. You achieve a stable, non-irritating oil-water mixture using materials that are intrinsically part of the natural tear film anyway. And clinical studies strongly confirm this approach works. PNE-based lubricants have been shown to improve tear film stability and effectively alleviate DED symptoms, directly validating this surfactant-free strategy.
It really seems like the entire R&D pathway is unified, doesn't it? Whether it's a liposome or a PNE, the goal is always using natural chemistry to get the emulsification and stability needed, completely sidestepping that toxicity trap of synthetic surfactants.
That is the single most important takeaway from the new formulation science playbook. Yes, use what the body uses, or something very close to it.
Just to quickly summarize the nanocarrier landscape for everyone listening, if we compare the irritation risk profiles of these different systems, what really stands out?
Okay, if we look across the spectrum of these advanced delivery systems, drawing from Table 2 in our source materials, nanocarriers like liposomes, and also things called solid lipid nanoparticles, SLN, and nanostructured lipid carriers, NLC, these consistently carry a low irritation risk. Why? Because they rely almost exclusively on biocompatible physiological lipids, things like triglycerides or phospholipids. The building blocks themselves are safe.
Okay, low risk for lipid-based carriers.
Right. Now, in stark contrast, traditional nanomulsions, the ones relying on older tech, carry a high irritation risk, often moderate to high. And that's because they rely heavily on those synthetic surfactants we keep mentioning, like polysorbate 80, for example. The simple reality is that the choice of emulsifier chemically defines the comfort level, and the new R&D pathway is uncompromisingly focused on lipid structures that mimic biological components to avoid that irritation.
So we've chemically designed this tiny, perfect, non-irritating bubble using natural lipids. It sounds great on paper, chemically sound, but taking it from a beaker in a sterile lab to a product that can sit on a pharmacy shelf for months or years and actually work when people use it, that requires overcoming two massive engineering hurdles. First, structural fragility during storage, and second, once it's applied, it's very short retention time on the eye.
Absolutely. These engineering challenges often dictate commercial success, almost regardless of the chemical brilliance behind the formulation. Liposomes, while thermodynamically stable in concept, can be kinetically fragile in practice. They break down over time. First, let's tackle that structural instability issue. A phospholipid membrane, even though it self-assembles, is inherently delicate. It's fluid, not rigid. Over the course of months, especially with fluctuating temperatures during shipping or storage, this fragility leads to physical breakdown. We see aggregation, where the particles clump together. We see coalescence, where they actually fuse into larger particles. And we see leakage of any encapsulated active drug out of the liposome.
And all of those ruin the product.
All of them. They destroy the crucial particle size uniformity, which leads to discomfort and blurry vision, and obviously, the loss of the drug means loss of efficacy. The shelf stability is simply too low without some kind of reinforcement.
So what specific chemical reinforcements do formulators add? How do they turn that fragile natural membrane into something structurally sound enough for a commercial product that needs, say, an 18 to 24-month shelf life?
R&D employs several pretty robust strategies, often in combination. One very common addition is cholesterol.
Cholesterol, like the stuff we worry about in food.
The very same molecule, yes.
Oh.
Cholesterol is a sterol, and it naturally integrates itself into the lipid bilayer, fitting between the phospholipid tails. It acts kind of like molecular scaffolding. It rigidifies the membrane, increasing its stiffness, making it less floppy, and crucially, it decreases the membrane's permeability to water and small molecules. This effectively reinforces the wall of the liposomal bubble against physical stress and prevents leakage.
Okay, so cholesterol adds rigidity. What else?
We also carefully select the type of phospholipids used in the first place. Formulators often prefer using highly saturated phospholipids. Saturated fatty acid chains are straight, unlike unsaturated chains, which have kinks or bends in them.
Right, like saturated versus unsaturated fats?
Exactly. Those straight, saturated chains can pack together much more tightly and orderly within the bilayer. This tight packing naturally forms more rigid, more stable structures compared to membranes made with more fluid, unsaturated lipids. It's a foundational material science choice to enhance stability right from the start.
Makes sense. And then there's the famous industrial solution for keeping nanoparticles from clumping together in suspension. It involves adding polymer chains to act as a physical buffer.
That's right, that's where we use pegylation. This involves chemically conjugating chains of polyethylene glycol PEG to the surface lipids of the liposome. Typically, these are PEG chains with a molecular weight around 2,000 daltons. When these PEG chains are attached, they kind of fan out from the liposome surface into the surrounding water. They create this bulky, hydrated steric barrier.
Steric barrier, like little bumpers.
Exactly like microscopic bumpers or maybe fuzzy polymer hairs coating the particle. These hairs physically prevent the liposome particles from getting close enough to each other to overcome their natural electrostatic repulsion and aggregate or fuse. This steric hindrance, this physical blocking, is incredibly effective at improving long-term colloidal stability, keeping the particles nicely dispersed, even under challenging storage conditions like temperature cycling.
Okay, so cholesterol, saturated lipids, and pegylation solve the structural stability issue in the bottle. Great. But the second big challenge is retention on the eye. The eye is an extremely efficient flushing system, right? It's designed to wash away anything foreign very quickly. How do we keep this carefully engineered nanoparticle on the eye surface long enough for the drug to actually absorb? How do we increase that residence time dramatically?
This is where we move into more advanced surface engineering, specifically myco-adhesion strategies. And these are absolutely essential because you're right, the typical dwell time for an unassisted standard eye drop is measured in minutes. Maybe just 1 or 2 minutes before blinking and tearing wash most of it away. That's not long enough for efficient drug delivery.
So how do we make it stick longer?
We need to leverage the natural biology of the eye surface. The cornea and the conjunctiva are covered by a layer of mucus called mucin, and this mucin layer carries a net negative charge.
Okay, the eye surface is negatively charged.
Right, so the strategy is to apply a cationic surface modification to our liposome. We give the liposome a strong net positive charge. The idea is to promote powerful electrostatic adhesion. Think of it like static cling between the positively charged liposome and the negatively charged mucin on the eye surface. We call this mycoadhesion. It's essentially creating a temporary, electrostatically driven chemical Velcro that holds the particle there.
That sounds very effective. What are the key chemical agents used to create this functional positive charge? And what kind of performance boost do they actually provide?
The primary agents used are high molecular weight cationic polymers. The most prominent example is chitosan, CS. Chitosan is a natural polymer derived from chitin. Or, an even more effective derivative is trimethylchitosan, TMC, which carries a stronger positive charge. By coating the liposome with a thin layer of one of these polymers, we ensure it carries that necessary positive charge to stick to the mucin. And the results are pretty astounding, really validating the concept. Studies consistently show that chitosan-coated liposomes have a significantly prolonged retention time on the eye. One study mentioned in our sources showed up to a massive 4.8-fold increase in the mean residence time compared to the uncoated versions.
Wow, almost 5 times longer on the eye. That's huge for drug delivery.
It's a game changer for bioavailability, yes.
Okay, but here's the critical point. This feels like a potential paradox, and it's where the chemistry gets really nuanced. We just spent a good amount of time explaining that benzalkonium chloride, BAK, is toxic because it is a small cationic detergent. It has a positive charge and detergent properties. Now we are enthusiastically describing the use of a different positive charge from a cationic polymer like chitosan to achieve dramatically improved function. Isn't that just replacing one danger with another, or is there a fundamental difference here? Is it just the size?
That is the single most important conceptual nuance in this entire deep dive, and you are exactly right to flag it. It seems contradictory at first glance. The safety distinction relies entirely, 100%, on the molecular weight and the resulting chemical mechanism of action.
Okay, unpack that. Molecular weight and mechanism.
Right. The toxicity of BA comes from 2 key factors. First, its small molecular size. This allows it to readily wiggle its way into, intercalate into, and penetrate the hydrophobic core of the cell membrane. Second, its inherent detergent property. It actively strips lipids out of that membrane, disrupting its structure. It acts like a tiny chemical drill punching holes in the cell wall.
Okay, small size, detergent action equal toxic.
Correct. Now contrast that with high molecular weight polymers like chitosan. These molecules are massive compared to BAK molecules, orders of magnitude larger. They are simply too large and too structurally complex to easily penetrate cell membranes or act as detergents in that same destructive way. They can't get inside the membrane easily. Their effect is confined almost entirely to the superficial and electrostatic interaction with the mucin layer on top of the cells. They stick to the mucin for retention, but they do not significantly disrupt the delicate phospholipid bilayers of the underlying epithelial cells.
I see. So we're using the positive charge purely for its functional benefit, sticking to the negative mucin, but we're avoiding the toxicity that comes from cellular penetration and detergent action simply because the tritosin molecule is too big to do that kind of damage.
Precisely. We've cleverly leveraged a powerful chemical property, charge attraction, while carefully excluding the cytotoxic mechanism associated with small cationic detergents. And importantly, histopathological analyses looking at actual eye tissue after exposure confirm this. Formulations using TMC-coated liposomes show excellent biocompatibility with the corneal epithelium. We maintain cell health while achieving that dramatically improved retention. It's the best of both worlds.
That's a really elegant chemical solution, but
Yeah.
despite this clear technical superiority, you mentioned there's a serious commercial and regulatory hurdle blocking these mucoadhesive agents like chitosan from widespread use. What's the roadblock?
Yes, it is a significant barrier to translating this excellent science into everyday products. Chitosan is, as I said, a natural, highly effective biopolymer. It even holds GRAS status, generally recognized as safe in many other applications, particularly in food and dietary supplements. People eat it.
So it's safe in food, but not approved for eyes.
Currently, yes. It faces limited, if any, specific FDA approval for widespread ophthalmic use. Because this mucoadhesive application represents a new chemical delivery strategy for the eye, regulators, quite rightly, require extensive long-term clinical tolerance studies. They need to prove its safety beyond any doubt, specifically in this sensitive environment, used potentially daily for years. This need for prolonged, expensive safety data collection means that translating this R&D excellence into mass-market, clinically approved eye drops is currently a slow and costly process. It remains a significant commercial barrier despite the obvious technical advantages of chitosan and its derivatives.
Okay, so let's assume we've designed this highly sophisticated, stable, and sticky nanoparticle. The chemistry is perfect, the safety profile looks good in the lab. The next massive challenge is logistics and process engineering. How do we actually manufacture this stuff reliably, consistently, in huge batches needed for the market? And then how do we get it onto the eye in a comfortable format that preserves the integrity of that delicate nanoparticle structure all the way to the patient?
Exactly. The manufacturing step has to preserve everything the formulation scientists work so hard to achieve chemically and physically. And critically, every delivery format, whether it's a spray or a gel, must maintain that preservative-free mandate, which itself requires specialized packaging like multi-dose preservative-free bottles or single-use vials and strict engineering controls during filling to prevent contamination.
Right, the preservative-free aspect adds complexity everywhere.
It really does. So let's look at the physical formats, starting with ophthalmic sprays. Sprays require a really complex formulation balance. They need to have a low viscosity so they can be easily atomized by the spray nozzle and feel comfortable during application. You don't want a thick jet hitting your eye. But they also need adequate viscosity once they land on the eye surface to ensure a longer dwell time, otherwise they just evaporate or run off too quickly. We talked earlier about how the solution to this is using materials with pseudoplastic flow behavior or shear thinning properties.
And how is that pseudoplastic flow typically achieved in these spray systems? What chemicals do they use?
Often it's through adding specific polymers, like hydroxypropyl guar gum or HPG. HPG is a type of polymeric hydrogel former. During the high shear stress experienced inside the spray nozzle or during the rapid shearing force of a blink, the long polymer chains in the HPG system align themselves temporarily, reducing the fluid's resistance to flow. This leads to low viscosity, the fluid acts thin, which minimizes irritation during application and blinking.
Okay, so it sprays thin, but then it thickens.
Exactly. The moment the fluid is static on the eye surface between blinks, the shear stress drops dramatically. The HPG polymer chains then rapidly begin to re-entangle and hydrate, absorbing water from the tears. This causes the system to gelify right there on the ocular surface. This in situ gelification dramatically extends the retention time, potentially from minutes to much longer, providing that necessary balance. Comfort during application, persistence afterwards. And sprays, specifically lipid-containing sprays, are proving incredibly effective for evaporative dry eye disease. They can quickly and comfortably spread across the eyelid margins and the tear film surface, replenishing that crucial lipid layer very efficiently.
That makes sense for sprays. Now, moving to the other end of the spectrum, what about formats designed for maximum retention, like gels and ointments? These are often used overnight, right?
Yes, gels and ointments inherently offer the longest retention time due to their high viscosity. The major formulation challenge here is ensuring that the fragile liposomes, or the PNE nanoparticles, remain structurally stable when they're incorporated into that very high concentration of hydrogel formers in a gel, or the greasy fatty base in an ointment. High concentrations of some polymers or oils can sometimes destabilize lipid particles if you're not careful.
So how do they protect the nanoparticles in these thick formulations?
The sources detail that a common strategy involves adding specific oil components directly into the liposomal suspension before mixing it into the gel or ointment base. Oils like mineral oil, maybe oleic acid, or medium-chain triglycerides like miguelate-12 are often used. This achieves several goals simultaneously. It helps stabilize the liposomal structure against the potentially disruptive environment of the concentrated hydrogel formers. And second, once applied, these added oil components contribute to the high viscosity and also form an effective anti-evaporative barrier on the eye surface, which is great for overnight use. The final product is essentially a complex, highly structured nanoemulsion within a gel or an ointment containing these protected active liposomal nanoparticles.
Okay, that covers the formulation formats. Now let's shift focus to the factory floor industrial scaling. We can make perfect nanoparticles using fancy lab techniques, maybe a few milliliters at a time, but if the method can't produce tons of this stuff uniformly and relatively cheaply, it's commercially useless, right?
It's exactly right. Commercial viability hinges on scalable manufacturing. The process must achieve two critical things: high throughput, making lots of it quickly, and guaranteed particle size uniformity, keeping everything consistently within that tight $2,200 textile range, batch after batch after batch. Traditional laboratory methods just cannot meet these industrial requirements. They're not designed for scale.
What's wrong with the common lab methods like ethanol injection or ether injection? They sound simple.
They are simple, but they fail commercially for multiple reasons. Take ethanol injection. It fundamentally limits the maximum lipid concentration you can dissolve, which means your batches are very dilute, small, and inefficient. Lots of solvent, not much product. Ether injection has its own problems. It requires relatively high temperatures, maybe 55 to 65 degrees Celsius, to evaporate off the residual ether solvent afterwards. This heat can easily degrade any heat-sensitive active ingredients you've encapsulated. And more critically, both of these solvent-based methods tend to produce particles with high size heterogeneity, what we call high polydispersity. You get a wide range of particle sizes, many outside the target range.
So you end up with particles that are too big, too variable in size, leading to poor comfort, blurry vision, unstable products, plus the potential risk of residual solvent toxicity. That's clearly unacceptable for an eye drop.
Precisely. That kind of variability, plus the solvent exposure and the heat exposure, are completely unacceptable for highly sensitive ophthalmic products aiming for maximum comfort and safety. The industry needed and found a method that uses high physical energy, not chemical solvents or high heat, to achieve homogenization and size reduction reliably at scale.
Which brings us to the industrial workhorse technique. High-pressure homogenization, or HPH. How does this method dominate the industrial production of these advanced nanocarriers?
HPH is, without doubt, the most industrially significant method for producing liposomes and nanoemulsions today, primarily because it's highly scalable and very effective. How it works is conceptually simple, but technically demanding. It forces the initial coarse lipid dispersion, which might have large uneven particles, through extremely narrow apertures or channels under intensely high pressures. We were talking pressures in the range of hundreds to thousands of bar immense physical force. This massive mechanical shear, combined with forces like cavitation and turbulence within the homogenizer, literally rips the large lipid aggregates apart. It then forces them to reform into much smaller, highly uniform and stable nanoparticles within that desired 2200 millimeter range. And it allows for the efficient, continuous processing of large commercial scale batches.
Okay, so it uses brute force to get the size right, but you mentioned something else interesting, that HPH doesn't just reduce particle size, it actually improves the stability too.
Yes, and this is a key insight that goes beyond just physical size reduction. HPH isn't just a processing tool. It seems to contribute to the product's quality. Studies have demonstrated that the HPH process itself significantly improves the storage stability of the resulting liposomes, especially at room temperature, compared to liposomes made using non-homogenized methods or those traditional lab techniques.
Why would the manufacturing process itself make the particle more stable later on the shelf? That seems counterintuitive.
The hypothesis is that the immense pressure and shear forces inherent in the HPH process don't just break particles down. They help the lipids reassemble into a denser, more thermodynamically stable, and kinetically favorable particle structure. In essence, the process technique itself acts almost like a stability-enhancing excipient. It helps the phospholipid molecules pack more tightly and uniformly, creating a more robust particle that's less prone to leakage or fusion over time. This intrinsic stability enhancement is another major reason why HPH has become the gold standard for producing these sensitive, high-quality nanocarriers at industrial scale.
Okay, one last manufacturing point. For maximum multi-year shelf stability, especially for products like sprays that might need to be shipped globally under varying conditions, sometimes removing the water entirely is necessary, right? To make a powder. How do they do that without completely destroying the delicate liposomal membrane structure during dehydration?
That requires specialized post-processing techniques, most commonly lyophilization, which is freeze-drying. Sometimes spray drying is also used. These processes convert the liquid liposome suspension into a stable, dry powder. This powder can then be stored for a long time and reconstituted, perhaps by the pharmacist or even the end user, by adding water just prior to use.
But freeze-drying sounds harsh. Doesn't removing all the water damage the membranes?
It absolutely can. The dehydration phase and also the freezing phase before it can cause massive mechanical and osmotic stress on the phospholipid membrane, leading to rupture, fusion, and particle failure. The structure collapses. To counteract this, these processes absolutely require the use of cryoprotectants or lyoprotectants. These are typically specific sugars, like trehalose or sucrose, or sometimes polymers. They're added to the liposome suspension before freezing. During the freezing and drying process, these molecules integrate with the liposomal surface. They essentially replace the hydration shell of water molecules around the lipid heads, forming a kind of glassy matrix that supports the membrane structure. They maintain the structural integrity of the membrane during the harsh dehydration phase. This ensures that when you rehydrate the powder later, the particles redisperse properly and are still perfectly intact, uniform in size, and fully functional. It's critical for powdered formulations. #6. Enhanced efficacy and future R&D horizons.
So the ultimate payoff of all this incredibly complex chemistry, this high-precision engineering, this careful manufacturing, it isn't just about making a drop that feels comfortable, is it? It's also about significantly enhancing the biological efficacy. We're building a highly sophisticated delivery vehicle that gets the active drug exactly where it needs to go within the eye. Much, much better than a simple aqueous solution could ever manage.
That's the entire point of developing these advanced nanocarrier systems, yes. Comfort is key for compliance, but efficacy is the ultimate goal. The liposome, or the PNE, acts as a highly effective Trojan horse, if you like. They fundamentally enhance the transocular penetration of drugs into deeper ocular tissues. Why? Because their lipid structure interacts much more favorably with the lipid-rich cell membranes of the cornea and other ocular barriers compared to simple water-based solutions. They can fuse with or pass through membranes more easily.
And we have proof of this improved penetration.
Oh yes, we see this very clearly in preclinical studies. Let's take an example. Delivering an antiviral agent called ganciclovir, or GCV, which is used to treat eye infections. Researchers consistently find that the drug concentrations achieved in various deep ocular tissues, places like the retina and the vitreous humor at the back of the eye, were measured to be anywhere from 2 to 10 times higher when the GCV was delivered via a liposomal formulation compared to delivering the same drug in a traditional aqueous solution.
2 to 10 times higher concentration in the target tissue. That's a massive improvement in potential effectiveness.
It's a huge difference. The nanocarrier system is simply much better at overcoming the natural barriers of the eye to get the drug where it's needed.
And you mentioned earlier that the size of the nanoparticle isn't just critical for comfort and avoiding blurriness, but it actually dictates where the drug is absorbed and where the highest concentrations end up within the eye.
Absolutely critical, yes. We've learned through numerous studies that the absorption route and tissue distribution are significantly size dependent. For instance, studies have shown that liposomes around the larger end of our range, say close to $200 text, seem to show preferential absorption via the scleral route.
Through the white part of the eye.
Exactly. penetrating through the sclera allows them to achieve the highest drug concentrations in the sclera itself and potentially reach the posterior segment, the back of the eye. This is vital if you're trying to treat diseases affecting the retina or choroid. Conversely, smaller particles, maybe closer to $50 to excess, tend to be absorbed more readily via the cornea, the clear front part of the eye. So, formulation chemists can actually tune the particle size distribution during manufacturing to preferentially target specific tissues within the eye, depending on the disease being treated.
That's incredibly sophisticated targeting just by controlling size.
It is, and furthermore, future R&D is pushing into even more advanced territory. We're starting to see work on systems like pH-sensitive liposomes. These aren't just passive carriers anymore. They're designed to be environmentally responsive. The idea is they maintain their integrity, they stay stable at the neutral pH of the tear film, around 7.4. But once they are internalized by a target cell, perhaps taken up into an endosome, which has a slightly more acidic internal environment, lower pH, that change in pH triggers a conformational change in specific lipids within the bilayer. This causes the liposome to become unstable, fuse with the endosomal membrane, and efficiently release its encapsulated drug contents directly into the cell's cytosol.
Wow, like a tiny smart bomb releasing its payload only inside the target cell.
That's a good analogy. It drastically boosts the cytoplasmic bioavailability, getting the drug exactly where it needs to work inside the cell, which is crucial for many intracellular drug targets.
Okay, looking at the overall clinical promise then, how do these complex, meticulously engineered, preservative-free systems really stack up against the older, traditional solutions, specifically for those chronic, debilitating issues like dry eye disease, DED, and meibomian gland dysfunction, MGD?
The clinical advantages are quite comprehensive, and they aim to address the underlying etiology of the disease more directly, not just masked symptoms. First, and foundational, the preservative-free nature minimizes the cytotoxicity we discussed extensively earlier. This demonstrably protects and supports the health and viability of the corneal epithelial cells over the long term. That's crucial for chronic use. Second, these lipid-based systems, both liposomes and PNEs, naturally provide hydration, yes, but critically, they actively support and replenish the tear film's crucial lipid layer. Since a deficient lipid layer is the root cause of evaporative DED-MGD, these formulations are treating the dryness while simultaneously fixing the underlying lipid deficiency.
So symptom relief plus addressing the cause.
Exactly. And third, they enable the targeted and protected delivery of specific active pharmaceutical ingredients. The sources mentioned, for example, the successful encapsulation of agents like curcumin. Curcumin is a potent anti-inflammatory agent, but it's highly lipophilic, hates water, and degrades quickly. The liposome protects the curcumin from premature degradation in the tear film and delivers it efficiently to the target tissues on the ocular surface. This helps to mitigate the chronic inflammation mechanisms that perpetuate these dry eye diseases.
That sounds incredibly promising. As we look toward the future then, what are the next R&D horizons? Where does this highly sophisticated formulation science go from here? You mentioned hybrid systems.
Yes, the next major push seems to be toward developing even more complex hybrid nanocarrier systems. Researchers are actively starting to integrate the distinct benefits of solid lipid nanoparticles, SLN, and nanostructured lipid carriers, NLC, with the more traditional phospholipid liposomes.
SLNs and NLCs, what are they, briefly?
SLNs and NLCs are similar to nanoemulsions, but they use lipids that are solid at room temperature, like solid fats or waxes, instead of liquid oils. NLCs are a more advanced version with a less ordered solid core. The advantage is that these solid lipid cores generally offer superior physical stability compared to the fluid membrane of a liposome, and they can often provide better drug loading capacity and more controlled drug release profiles.
So combining them with liposomes gives you...
The idea is to create a hybrid structure that potentially gives you the best of all worlds. You might get the enhanced physical stability and drug reservoir capacity from the solid lipid core, from the SLNLC part, combined with the excellent biocompatibility, the cell interaction properties, and maybe the surface functionalization capability of the outer phospholipid shell from the liposome part. It's about synergy.
Very interesting. And what about biomimicry? Does the future lie in just using general phospholipids like PC, or in achieving an almost perfect replica of the natural tear film's complex lipid mixture?
The future definitely lies in pursuing ever more precise biomimicry. Current advanced liposomes use general, high-purity phospholipids like PC or PE, which is already a huge step forward. But the natural human tear film lipid layer is incredibly complex. It contains dozens of different lipid species in very specific ratios. Future formulations, the truly next-generation ones, will likely need to increasingly mimic this natural tear film lipid profile with near-perfect accuracy. That means achieving not just the right types of lipids, but the exact physiological ratios of things like sphingomyelin, SM, phosphatidylethanolamine, PE, phosphatidylcholine, PC, cholesterol esters, ceramides, and so on.
Getting the recipe exactly right.
Exactly. Achieving that precise physiological lipid recipe will likely guarantee the best possible stability of the reconstituted terafilm layer after application, and crucially, it should elicit the absolute best, most natural physiological response from the patient's ocular surface, minimizing any remaining potential for incompatibility.
And finally, let's circle back one last time to what seems like the biggest translational challenge right now, that regulatory frontier for the best performing additives.
Yes, unfortunately, the largest ongoing challenge, the main bottleneck holding back some of the most promising technologies, remains that regulatory frontier. We have the clear technical excellence, especially with those mucoadhesive agents, the high-performing cationic polymer coatings like Titosan and TMC. We know they work incredibly well. They offer that dramatic, proven 4.8-fold improvement in retention time, which would revolutionize treatment efficacy. But translating this technical superiority into widely available, mass-market, clinically approved products requires generating that extensive long-term clinical safety data package that regulators demand. And generating that data currently acts as the single biggest commercial barrier that the field must overcome in the next decade of ophthalmic formulation science to bring these truly advanced therapies to patients. #Tag7.
That truly brings us to the end of a deep and, I have to say, fascinatingly technical dive into the future of eye drops. We started by defining the problem at a molecular level, didn't we? Moving from the specific pain caused by simple detergents activating that TRPV1 pain receptor, flooding cells with calcium, all the way to the sophisticated, self-assembling natural chemistry of phospholipids forming liposomes and PNEs. We then explored how advanced chemical engineering steps in to solve those crucial practical problems, enhancing structural stability using things like cholesterol and pegylation. And now we can dramatically increase retention time using carefully chosen charged high molecular weight polymers like Titosan, all while meticulously constantly avoiding cellular toxicity.
What this whole journey demonstrates, I think, quite powerfully, is that the next generation of comfort medicine, particularly in eye care, is fundamentally driven by a strategy of chemical exclusion, maybe even more than chemical addition. The goal isn't just to add better ingredients. It's really about systematically eliminating every potentially toxic or irritating synthetic component, every harsh surfactant, every traditional preservative, and replacing them entirely with highly targeted biocompatible nanoparticles that are designed to act almost like natural tissue components. It's a perfect demonstration, really, that sometimes the most sophisticated and advanced design is the one that most closely imitates and actively supports nature itself.
That's a great way to put it. So here's a final provocative thought for you, our listeners, to consider as we wrap up. Considering the powerful, the undeniable, and frankly the necessary potential of these high molecular weight cationic polymers like chitosan for dramatically improving ocular drug retention, how might R&D strategists or perhaps even regulatory bodies work to accelerate their acceptance specifically in the ophthalmic space? Could one approach be to focus first on achieving a more limited approval, maybe for formulations used in non-penetrating applications initially, perhaps for things like lubricating post-operative surfaces or treating external eye conditions where long-term daily use isn't the primary scenario? Could demonstrating safety in those contexts build an undeniable long-term safety record that eventually allows for broader approval, paving the way for their use in the more widespread critical chronic eye treatments like DED and MGD, where their benefits are so desperately needed? The chemical path forward seems clear and incredibly promising, but navigating that regulatory pathway remains perhaps the steepest climb for commercial viability. Something to think about.